Hello, this begins the Unit 2, Module 4, Chapter 7, Part B presentation. The big picture for this presentation is that we're going to be talking about transcription in some detail, translation, and then the differences in these processes between prokaryotes and eukaryotes. Let's discuss transcription. Transcription is the first stage of gene expression involving RNA polymerase using a template DNA strand and only one of those strands, the coding strand. RNA polymerase binds to the promoter region. It uses no primer. It proceeds with transcription until it reaches a terminator sequence on the DNA, and then it stops. And the process concludes with an mRNA transcript. Like replication, messenger RNA synthesis proceeds in the 5' to 3' direction, but only on one strand. So the resulting mRNA is complementary to a negative DNA strand and has the same sequence as the positive DNA strand, with uracil instead of thymine. If the mRNA carries only the information for one gene product and one protein, it is called monocistronic. Polycistronic messenger RNAs code for more than one gene product or protein on a messenger RNA molecule. RNA polymerase needs guidance on where to begin messenger RNA synthesis on the DNA. Sigma factors are components of the RNA polymerase enzyme that allow the enzyme to recognize promoter elements that are transcription initiation points on the DNA. Transcription, or RNA synthesis, is described in 3 basic steps: initiation, elongation, and termination. Bacteria use sigma factors to direct RNA polymerase, while eukaryotic cells use transcription factors to recognize promoter sequences. A promoter is the region of DNA that initiates transcription of a particular gene. Promoters are located near the genes they transcribe on the same strand and upstream on the DNA, toward the 3' region of the antisense strand, also called the template strand. Remember, RNA is synthesized in the 5' to 3' direction. The major components of transcription are summarized on this table 7.3 from your textbook. The genetic code defines how sequences of the nucleotide triplets, called codons, specify which amino acid will be added in order during protein synthesis. With some exceptions, a 3-nucleotide codon in a nucleic acid sequence specifies a single amino acid. DNA sequence defines the coding region of a gene from the beginning until the end of the region to be transcribed. A codon is defined by the initial nucleotide from which translation starts. Every sequence can, thus, be read in 3 reading frames, each of which will produce a different amino acid sequence. The actual frame in which a protein sequence is translated is defined by a start codon, usually the first AUG codon in the messenger RNA sequence. AUG specifies the amino acid methionine. The ribosome is the complex molecular machine found within all cells that is the primary site of translation. Ribosomes link amino acids together in the order specified by the sequence of the messenger RNA molecules. Ribosomes consist of two major subunits. The small ribosomal subunit reads the messenger RNA. The large subunit joins amino acids to form the polypeptide chain. Each subunit is composed of one or more ribosomal RNAs, or rRNAs, and a variety of proteins. A transfer RNA is an adapter molecule composed of RNA that serves as the physical link between the nucleotide sequence of nucleic acids, DNA and RNA, and the amino acid sequence of proteins. tRNA does this by carrying an amino acid to the ribosome, as directed by the 3 nucleotide sequence codon in a messenger RNA molecule. tRNAs, therefore, are a necessary component of protein translation. Because there is no nuclear membrane separating messenger RNA transcription from the ribosomes in prokaryotes, translation begins before transcription finishes the messenger RNA molecule. The next few slides detail the steps of peptide synthesis by the ribosome. The process begins with the ribosome recognition of the initiating AUG codon for methionine to be the first amino acid in the chain. The next amino acids are added as the ribosome moves to each successive codon, waiting for the proper tRNA to diffuse in and hydrogen bond with its anticodon to the codon of the messenger RNA. The next amino acid is covalently linked to the previous amino acid, and the ribosome moves to the next codon on the messenger RNA. AUG begins the chain. Diffusion and hydrogen bonds are the keys. The ribosome links the successive amino acids via peptide bonds. The ribosome moves along the messenger RNA in the 5' to 3' direction. And the process continues, elongating the specified protein. The messenger RNA is finite. It had a termination sequence in the DNA to end transcription, and there is a signal in the messenger RNA to end translation. The signal to end translation is a combination of any 3 codons which do not specify an amino acid, namely UAA, UAG, or UGA. Translation will end because there are no tRNAs with anticodons capable of making hydrogen bonds with any of those 3 codons. The stalled ribosome will disengage from the messenger RNA and translation ends. Once translated, proteins undergo further manipulations to assume their final functional state. These modifications include proper folding with assistance from specialized protein facilitators called chaperones. These proteins, ready to be carried across the plasma membrane, are already predestined by an amino acid signal sequence to direct that outcome. A hydrophobic set of amino acids makes up the signal sequence, and this sequence ensures the recognition of those proteins and their proper transport. Eukaryotic transcription and translation are hugely more complex in that messenger RNA is modified in many ways, prior to translation. These extensive modifications, called processing, allow for further control on the eventual protein structure and output from eukaryotic genes. The modifications include taking the raw messenger RNA, called pre-mRNA, and splicing to remove non-coding introns and linking coding exon regions in a protein complex called a spliceosome. Then covalently linking a modified nucleobase to the 5' messenger RNA end. This is called capping. And then finally covalently adding a series of adenine nucleobases to the 3' end, a so-called poly-A tail. Each of these processes is a control point for eukaryotic gene expression. Here is a summary of eukaryotic post-transcriptional mRNA modifications. Ribosomes from bacteria, eukaryotes, and archaea differ in their size, sequence, structure, and the ratio of protein to RNA. The differences in structure, as we will soon see, allow some antibiotics to specifically kill bacteria by inhibiting their ribosomes while leaving human ribosomes unaffected. In bacteria and the archaea, more than one ribosome may move along a single messenger RNA chain at one time, each reading its sequence and producing a corresponding protein molecule. The ribosomes in the mitochondria of eukaryotic cells functionally resemble many features of those in bacteria, reflecting the likely evolutionary origin of mitochondria. This slide contains questions to reflect upon and use the presented information.